Maybe the ones that are like all of these represent all of them.
Yes.
Gosh, I messed up with all of them.
I was like, it's fine.
I think just one. Not all of them.
This is a trick.
So as I said, guys, this, right, chapter 12, part 2 recorded lectures there, and we're doing part 1 right now. So we have everything for the next exam. 10, 11, and 12 are all covered for after today. All right. Oh, I need the PowerPoint. Okay, so it's the chapter 12 lecture. Like I said, if you want to open it up and follow along, you can on your own, but I have my screen shared here. So interrupt me, those of you guys on Moodle, if you have a question. Oh, no, Mariana already talked. I know I can hear you because I did that in my other class with my freshmen. I had them like muted. And I was like, oh, nobody's gonna tell you that. I'm like, nobody's gonna answer on Zoom. I was like, all right, whatever. And then they're like, oh, somebody texted somebody. They're like, the sound's off. And I'm so sorry. Sorry for being a jerk. But anyway, it was like 3/4 of the way through class though, at that point. I was like, all right. Anywho, I can hear you guys on Zoom. I can hear you guys in class. So interrupt me. I feel like this chapter for you guys, you bio majors, might not be so bad. I don't know. I feel like this exam, maybe a lot of this stuff, do you guys agree? Maybe not the inflammation chapter, but I feel like chapter 10 and chapter 12 are a lot of the concepts are things that we've already talked about. But whatever. It's always good to have a refresher. There's definitely some new stuff too, but anywho. I'm recording, right guys? On Zoom? Recording? You can see my screen? All right. Like I said, interrupt me. You know, this won't be 3 hours of lecture on biochemical molecules, so don't worry. Okay, so neoplasia. So chapter 12, obviously we're focusing on overgrowth of cells, right, in cancer, okay? So first one, we got to talk about all the concepts that are related to this, just like we do in every chapter. Sorry for the weird font. I thought I had changed that. Okay, so first, just, you know, some definitions that we should be familiar with. So neoplasia, right? That's just abnormal growth, okay? It doesn't necessarily have to be cancerous at that point. So tumor, or you can call it a neoplasm, again, is just this abnormal growth of cells or a sort of collection of an abnormal growth of cells. Then we can talk about benign versus malignant. Again, we probably know all these terms. Yes, we're all familiar with these. Okay. If you just like define that term cancer, it says it encompasses about 200, more than 200 diseases, right, that occur at different ages with different rates of growth, differentiation. It says detection evasiveness. I don't know. What do you think that means? I mean, if you think about different types of cancers, what are some things that are going to differ between those different types? That's essentially what this is listing here. What do you guys think? What are some things that differ? We talk about greater than 200 different types of cancers that exist, right? So what's going to be different between one versus the other? Give me some possibilities. Size, what do you mean? Like size of the tumor? Well, think about just like different types of cancers in general, right? Even if it was 2 cancers that both form tumors, what are we, when we talk about different types of cancers, how do we categorize them, I guess? Based on what?
Like if it's spreading or not.
What it affects? Yes, stages, true, right? We can grade them by stage 1 to stage 4 in terms of seriousness, but
the type of organ.
No, you did. What do you mean? You know what you mean.
The nervous system.
Yeah, what's this, right? Organs, lung cancer versus liver cancer, right? That's what you meant, yes? Okay. Somebody on Zoom was going to say something. What were you going to say?
I just said the type of organ, but you.
Yeah, type of organ. Oh, see, look, my volume did go down. I swear I turned this all the way up. What else? Somebody else on Zoom. I don't know. What are some other differences that?
Like
Rachel.
whether it's spreading or not.
Yeah. Okay. Yeah. All right. What else? What does differentiation mean there? So I think we kind of
I
hit,
guess.
go ahead.
Oh, I was gonna say, I don't know if this is right, but I was gonna say like whether or not it's like it's killing cells faster versus like if it's just rapidly like dividing. I don't know. That might not be right. It's fine.
Well, that's not really right in terms of what differentiation means, right? Think about differentiation. What do we mean by cell differentiation? Because this does relate to cancer.
Look different compared to like normal cells.
Nope, not really.
So
Or when
isn't
the
that like
cells
when they?
grow.
Oh, go ahead, Fabi.
Oh,
Fabi, go ahead.
when the cell is growing, it differentiates depending on where it's dividing. So if it's in the lungs, the cell is gonna turn into lung tissue, and if it's in the brain, it's gonna turn into brain tissue.
Okay. Yeah, so you're on the right track, right? The idea of differentiation is what the cell is going to become specifically, right? So once cells are differentiated, they become a lung cell or a neuron. At that point, when they're differentiated, they usually don't have the ability to divide, right? So when we talk about cells, again, lung cells, heart cells, whatever, those are fully differentiated cells that have a specific function. What happens with cancer cells is they sort of de-differentiate. So they sort of back up into the stem cell state, and then that's how they can divide. So you lose differentiation when you talk about cancer cells, okay, and stem cells. So we'll talk a little bit more about that, but let's definitely be able to define what we mean by differentiation. That probably will come up again, hint, hint, because I feel like we get tripped up on that, right? Like we all kind of got tripped up on what we mean by that, okay? So make sure you understand that concept. And like I said, it'll come back again as we move through the lecture, okay? When a cell is differentiated, fully differentiated, it means it's in its final mature state, right? It has its specific function, like a neuron, like a cardiac muscle cell, okay? Again, cancer cells sort of lose that differentiated state and are in a stem cell state, and then they are able to proliferate, okay? Where they should really remain differentiated, but they don't. There's all sorts of cellular changes that cause them to be in this weird kind of de-differentiated state. Okay, questions about that? Like I said, that'll come up again, okay? All right, I guess we can move on. But I think the one big thing is that let's remember that there are so many different types of cancers, right? And some are treatable, others aren't, right? Some you can treat with surgery, others only with drugs, right? There's so many differences in terms of those cancers and in terms of how you would treat them and how they form and what have you, right? So just remember how complex, sort of this topic is.
Oh, also,
Okay.
I think
Yeah.
I remember, like it was something about I can't remember the word exactly. I think it was like
Metastasis?
Metastasis.
Yeah, where like it would be like the direct blood, and that would
Yes.
make it so.
Yeah. We'll talk about that too. Yeah. Angiogenesis, are you thinking about that? Or no? Well, we'll talk about that.
Yeah, I was also thinking of that.
All right, so we'll talk about all those concepts as we move down. This is just meant to kind of get us thinking about some of these concepts that are related, but we're going to talk about them more. Okay, so genetics, right? Genetics is involved in cancer. I'm sure we would all say that, yeah? Are cancers fully genetic disorders? Or is it only genetics that's involved in sort of causing cancer or forming cancer? No, right? Definitely not, right? There's definitely this multi-hit hypothesis to cancer, right? There's a genetic predisposition, there's genetic changes that occur, but there's also environmental factors that come into play as well, okay? And again, we'll talk about this a little bit more. Just to remind you, yeah, mutations are gonna occur in cancer cells, right? That's DNA damage. Different types of mutations though. If you say something is a germline mutation, okay, it means that these are mutations that are inherited, right? That you basically got from your mother or your father. This is from birth, right? This mutation was already there in all of your cells. Does that make sense, right? Okay. However, there are mutations that will occur like throughout your life, right? Like UV light can cause damage to your skin cells, right? Mutations that then could lead to cancer, okay? Those are somatic mutations. Those aren't mutations that were there already. There's some sort of agent that induces that genetic change or that change in the DNA. Does that make sense, everyone, the difference? I'm not sure I love this drivers and passengers terminology, but we'll just go with it. It's just in case you guys kind of hear it again. The book uses this terminology. The idea that drivers are mutations that basically we know can cause cancer, right? They are mutations in tumor suppressor genes, right? And these are targets for cancer therapy, right? We know that if there's a mutation in a tumor suppressor gene and that gene's not able to function, it's going to lead to uncontrolled cell division, right? And proliferation, okay? However, there are also other mutations that kind of just happen along with these, that they're just mutations that you may often see, but they're not necessarily connected to a tumor suppressor gene or some other cancer-related gene, okay? So it says they just accompany the drivers. So for whatever reason, when you see the mutation in the driver in the tumor suppressor gene, you also sometimes see these accompanying kind of mutations, if that makes sense, okay? These passenger mutations aren't going to be like a therapeutic target, okay? Like I said, they kind of just come along for the ride. That's the name passengers. Does that make sense to everyone? So again, they may be mutations that we see in cancers, okay? Like if you kind of looked at association of mutations between different types of cancers, you may see these more often than not. However, again, they're not really related or they're not a mutation in any sort of cancer-related gene, okay? They're just there because they happen along with those other ones. Questions about that? Let me see, I'm gonna switch my mask. I brought a thinner mask. I knew it was gonna be annoying, so this is why I don't lecture in person. Let's see if this is better. Yeah, I use this mask. I use this when I was when I teach fitness classes too. So anyway, it makes a big difference. Okay. So all right, so then this is just saying, well, if you have a mutation, right, in the DNA, what does that then lead to? I mean, it's going to lead to gene products that are not correct, right? Potentially. And then those can result in changes in cell division, right? Proliferation. They can help with angiogenesis, right? Adding more blood vessels to the area where the tumor is. That's going to make those cancer cells able to keep proliferating, okay? And then why is this weird thing here? Can I close this thing? Yeah, I think that does close it. I think I did that before. Alternative form of energy production, right? These are all kind of these altered DNA sequences that you see in cancer cells, you know, lead to changes like this. That then gives those cancer cells the ability to keep dividing, right? And doing all these weird things that they shouldn't normally do, okay? So why would the alternative form of energy production be important? And it mentions there anaerobic glycolysis. I guess you can kind of think of maybe this kind of should have been first, right? If you think about like when a tumor's first forming, and maybe they don't have, those tumor cells don't have the blood supply that they necessarily need to be able to keep dividing like that, they will go through sort of alternative forms of energy production, right? Like glycolysis to be able to produce the energy that they need at that time to keep dividing. So they find ways to kind of like get around any sort of barrier, right? You know, if they couldn't do that, maybe those cells would have went off to die, right? Because they wouldn't get enough energy. But because they can sort of find this alternate way, they're able to survive when they really shouldn't, okay? All right, so here's our lovely figure that we always have in terms of relating these concepts. And I think we kind of talked about some of these already. Again, cellular regulation is in the middle there because we're talking about, oh, Rachel, you got a question?
Yeah, I did. I just
Yeah.
wanted to know, like, what
Thank
is
you.
it called? Like when the tumor or those like cancerous cells just kind of make their own like vascular system? 'Cause I know you were-
Angiogenesis.
What's that?
Angiogenesis is listed here, see it?
Oh, okay, okay. Thank you.
Yeah, and we'll talk about that again too. Other questions, guys? All right, so, oops. All right, so if we take a look at this figure here, again, cellular regulation is in the middle because we're saying that cancer cells, right, are misregulated, right, or deregulated. They're not regulated at all. They're basically overriding these regulatory mechanisms and they're able to divide, okay? If you look, obviously, you know, if there's problems with cellular regulation, it could then lead to increases in reactive oxygen species, right? Inflammatory molecules in general can lead to oxidative stress. That itself can cause more genetic mutations too, right? So that's where that's kind of going. Again, if you think about stress and immunity kind of all linked, again, that's the idea of inflammation, okay? And that again, more inflammatory, like we just talked about in that last chapter, right? If there's more inflammatory molecules around, it can damage cells, right? And then potentially can put them in this cancer state as well. All right, things in the environment obviously can then be triggers to sort of continuing this sort of cancer process, okay? So a cell that might kind of be misregulated a little bit and maybe has a mutation or something, if now it's encountering, you know, chemicals from smoking, right? Or some other environmental allergen that may then push it to go further down that sort of route, okay? All right, and then it has mood and affect over there as well, and that, you know, deregulation of cells can also affect that as well, right? And then potentially these then can sort of influence kind of and work back on, again, sort of increasing properties that would make cells more cancer-like, okay? All right, let's move on here. Okay, so a little bit more about benign versus malignant, okay? We already talked about this a little bit. And again, sorry, I thought I got rid of all the weird italic font, but apparently I did only with some of it. Okay, so benign means what? Cancerous or not?
Non-cancerous. Not.
Not cancerous,
Cancerous.
okay? Typically they'll develop slowly, but not always. They're usually encased by this connective tissue capsule that keeps it localized, right? If we're gonna name these, they will end in a suffix like oma, right? Adenoma, right? Instead of down here, if you look at carcinoma, right? Or sarcoma, right? That's gonna indicate cancerous, okay? But this is a medical term. You can learn that more medical term. But anyway, non-threatening usually, but sometimes a benign tumor can be threatening, right? If it's going to push on something, like especially in the brain, you know, if it's protruding on some other surrounding brain tissue, right, and affecting the function of other cells, then it could potentially be dangerous as well. Okay. All right, and so that's what that's saying there. All right, now if we're talking about malignant, right, malignant tissue, okay, if you talk about it originating from epithelial tissue, okay, usually we call those carcinomas, okay? If it originates from parenchymal tissue, okay, it can end in sarcoma, okay? Bottom line is, I guess, for us right now, let's know that, like, if we just see like adenoma, that indicates that it's a benign tumor. But if you're seeing carcinoma, sarcoma, those are, you know, cancerous, indicating cancer, okay? We move on, right? If we're talking about hematopoietic or lymphoid tissue neoplasms, right? So these are blood cells, right? Leukemias, lymphomas, we've heard of those types before, I'm sure. Okay, I think that's kind of most of what's there. So in terms of, you look at the actual cell itself, right? Usually they have a weird, like, large nuclei. There's abnormal amounts of DNA, limited cytoplasm, and they look immature. So how does that relate to the differentiation concept that I talked about before? So, right, like, if you got a biopsy, what happens to that piece of tissue that the doctor takes? What do we do with it? What do you do with it in a biopsy? Well, you take, I mean, you could get like, if you, so after it's taken out of your body, right, a small amount of tissue, and it could just be like a little needle goes in, right, and pulls out a small amount. What do we do with it? How do we analyze it?
Like look under a microscope?
Yeah, look under a microscope, right? I mean, in clinical lab science, right, like, you know, people who are going to clinical lab science, that might be your job, right? You're in a clinical lab, you're looking at histological samples or tissue samples from patients. And what are you looking for? You're looking for some of these things that are mentioned here, right? You're looking for cells that look cancerous. It's by eye, it's not like a machine, that part, right? Now there are other types of markers and things like that that you can run through a bioassay, but for the most part, if you're talking about, you know, a biopsy, a tissue biopsy, somebody's looking under a microscope, right? And so again, that immature appearance relates to differentiation, right? Those cells in that particular tissue, right? We're assuming that we're looking in an area where they should have this mature appearance. Meaning, like, if you're thinking about a neuron, a lot of you guys are in neurobio, right? What does a neuron look like if it's mature, right? If we just talk about like a characteristic neuron that I always draw terribly on the board, right? It's got a cell body, it's got dendrites, a long axon and an axon terminal. It's very elaborate, right? In terms of all those, the structures of the cell. Well, an immature neuron doesn't look like that, right? It wouldn't even have any of those processes. And we talked about that in neurobio too, when we talked about like development, right? We talked about like a neuroblast. Okay, so it's the same idea. In different tissues, you know what a mature cell would look like. But if you see these immature cells, these dedifferentiated cells, that indicates that, you know, there's cancerous tissue there, cancerous cells there, okay? So all the more reason why we should be happy that we know how to use a microscope, right? I mean, you guys, even without any other training, as long as you know how to use a microscope and somebody trains you on what you need to look for, you could do a job like that, right? All right, so grows rapidly, invades surrounding tissues, right? And it says it will metastasize, right? And spread to other organs. So that's the thing, when we talk about something being malignant, we're basically suggesting that it has the ability to metastasize. Right, Rachel, what's up? Another question?
Yeah, I have another
Go ahead.
question.
Yeah.
So like when you said benign, it reminded me of like fibroids. So is like that different from like a benign like tumor?
No, same idea. Any benign growth, neoplasm is just abnormal growth, right? So really any growth could be considered a benign neoplasm, right? So any sort of abnormal growth of cells. Yeah, and you know what, usually when you say tumor, that usually does suggest cancer, right? So you may just say a benign growth or something like that. But technically, I guess if you said benign tumor, it would mean the same thing. Does that make sense?
Yeah.
It's a matter of terminology, I think. Okay, so this table just compares benign versus malignant. Okay, so we should be able to distinguish between the two. Okay. All right. So next learning outcome, compare and contrast cell cycles for normal and malignant cells. So sorry to bring back cell cycle, but obviously if you're going to talk about cancer, you have to talk about cell cycle, right? Why?
Because the cell cycle basically controls the cell division, cancer, and the cells continue to divide.
Yeah, right. So cancer cells are continuing to divide. We talk about cell cycle, cells that are in the cell cycle are dividing, right? If the cell's unable to control that cell cycle, things divide, right? When they shouldn't. All right. So some of this you guys know. Okay. Well, anything on here that's new to you? Probably not. Right? You probably know all this. At least you should from cell bio. It says one thing that I guess might be. Oh yeah, I forgot about that. The ligand band. Maybe you should bring them back. Bring back.
Oh, we're still back. We're still.
You're still in existence. The one thing that it's cancer cells do not necessarily have shorter cell cycles, it says. It's just that they'll move through that cell cycle many more times than a normal cell would, right? So it doesn't necessarily mean they have to have a shorter cell cycle. They're just moving through that more than other cells. All right, I think we should know all of this. Yeah, I got a picture, I think. Right, so we should know the phases of the cell cycle. And so for you guys, maybe you already know this. Yeah, maybe you forgot. And this is just a reminder. If you talk about, right, interphase versus the mitotic phase, right? If we want to talk about the cell cycle in general, there's interphase, the mitotic phase. In interphase, you have G1, S, and G2. We all remember that, right? And then phases of mitosis. We're not going to worry about phases of mitosis. Don't worry. Okay, I'm not going to like show you pictures and ask you about phases of mitosis. We leave that out. The other thing here, G0. I really should be. I guess all of these should be like a sub, like G sub 0. But whatever. What's G sub 0? Is that really kind of part of the cell cycle? You think?
Not really.
Yeah, not really, right?
No.
It's Natasha said not really, which is true. It's the cell sort of, when cells exit the cell cycle or in a sort of resting state, we say they're in G0, okay? So there are cancer cells, and I think I mentioned this in a couple slides, but there are cancer cells that can go into G0, right? And then if at that point you were assessing a patient for cancer, you know, it may not look like there is any cancer there, right? But then at some time, when there's some environmental agent or something like that, stress, inflammation, that then triggers those cells to re-enter the cell cycle, then you sort of have this exasperation, right? You know how people can go into remission from cancer, right? And so some of these cells will go into they're like tricky, right? They're like hiding. And then they'll enter back in and start causing problems, okay? So it's hard, it makes it harder to diagnose. All right, so here's a figure we should also be familiar with, okay? In red are the cell cycle checkpoints, okay? The hell is that green thing say? I don't know why it says antiphase. I guess that must be I'm assuming that's a typo. But maybe not. Maybe that's just a different name for something. I'll have to look that up. But bottom line is, we just want to know that we have these different, or remember that we have these different checkpoints. I'm not going to ask you guys to like explain the checkpoints because that's not what we're focusing on. We do that in cell bio because it's cell bio, right? But in here, we just want to be aware that we do have these checkpoints, right? That basically, you know, what's happening here? If we look at this checkpoint in red in S phase, what's that checkpoint checking for at the end of S phase there? Primarily, what's that checkpoint looking at? What happens in S phase?
It's to see if the DNA was like replicated, right?
Yeah, so right, DNA synthesis is going to happen there, right? DNA replication. And so really that checkpoint is sort of checking to make sure the DNA was replicated correctly. If there is some sort of DNA damage that's recognized, what will happen?
It will like try to fix it, or
Okay.
like if it can't fix it, then it'll induce like apoptosis.
Yep. Good. Correct. The cell will try to fix it. The cell does have mechanisms to fix, right? Mistakes. But if it can't, then it should undergo apoptosis. Well, what happens in cancer cells sometimes?
I think
Yeah, the checkpoint is like overridden, right? So like there might be DNA damage. That cell should go through apoptosis, but it doesn't, right? It allows that cell then to divide and keep producing more and more of these cells that have DNA damage, right? So basically in cancer, right, the regulation of the cell cycle is overridden. And so, and one of those ways is that these cell cycle checkpoints aren't really doing what they're supposed to do, right? Okay. All right, so there's my differentiation. I told you I was going to talk about it more. Let's see if I probably mentioned most of this already, right? So it says a blast cell or dedifferentiated cell is most immature. That's like a stem cell. So when you think about stem cell, what do you think about? Like, how would you define a stem cell? I always use the example of like your mom or whatever. Does your mom and like ask you questions about science sometimes? Or I don't know, anyone in your family ask you questions about science, like to define something? I don't know. What do you think about when you think about a stem cell, I guess?
Can it be like anything you want it to
Okay, good. It has the potential to really be anything. Yeah, right. If you think about embryonic stem cells, right there, you start off with one single cell, a whole bunch of proliferation happens. And then once you get a whole bunch of cells, then they start to differentiate into specific cell types. So yeah, stem cells can kind of be anything. And they also have this really great ability to divide and proliferate. Okay, so a blast cell is like a stem cell. Okay. All right, I already said this, right? They lose their differentiation, those cancer cells, and they appear to be very similar to blast cells or sort of stem cells. Okay, they have the stem cell-like appearance and behavior. Okay, if we talk about grading, right? So you can grade the level of differentiation. And remember, I just told you, right? Somebody's going to look at a tissue sample, the biopsy, right? And look at these cells and to see what they look like, right? To indicate if there's cancer there. And there is a little bit of a grading, and there's obviously other criteria that they're going to use as well, but this is an example of one. Okay, so it says grades 1 through 4. The lower numbers, and I have a picture in a second. The lower numbers refer to cancer cells that are well differentiated, okay, with specialized function. So which one, grade 1 or grade 4, is more serious in terms of cancer?
4.
Yeah, 4. Why? 4 is going to have what? Is it going to have less differentiation, grade 4? It says poor differentiation, right? Less differentiation, meaning a lot of the cells are going to be de-differentiated, right? And look stem cell-like. So that's going to indicate that there's probably a lot of cancer cells there, right? Okay. It says here grade X is used to describe tissue that can't be assessed. They probably then do another biopsy, right, from another area. Right, so there you go. You got well differentiated down to grade 4 there, where everything just looks a mess, right? What do those types of cells look like there? Reminds me of a particular cell type.
Like epithelial?
Yeah, epithelial. And which particular type of epithelial cell do you think? No, they're plump. Plump and kind of like squarish. Yeah, cuboidal, right? So they kind of look like simple cuboidal, maybe things that we'd see in kidney tubules, right? Anyway, this is what they should kind of look like right here. But then again, you can kind of see how they start to lose their shape. The nuclei are going to look weird. And then you basically sort of lose. So like if this was a tubule, like a kidney tubule, you're basically losing that structure too, right? They're losing their association with each other. Okay, so that's something too to kind of look at, or something that kind of indicates that they're not in their differentiated state. Okay, questions about that? All right, moving on. So how do genetic factors interact to affect the molecular changes associated with malignancy? So we got here. So yeah, so we've already started talking about some of these things, right? So let's think about, what's listed here, again, outside of the cell, factors that might occur that are then going to end up altering gene products, right? And then that will alter the function of the cell, okay? So it says environmental variables like oxidative stress, hormonal influences, lifestyle factors, chronic inflammation. I'm sure you can list other things as well, right? What are some particular, I guess, environmental variables or factors that you can think of that might be related to cancer? Okay, good. Yeah. Exposure to sun, smoking, things like asbestos, right? There's chemical exposures which they cause lung cancer, right? And before we were sort of aware of that, there are people that worked in, industry, and they really were exposed to asbestos every day, and now they may have, lung cancer. Okay, it says here, interactions of genes with these variables can initiate and promote the change from normal to then cancer. Okay, so the idea is that everything may be kind of fine until there is some environmental exposure, whether we're talking about asbestos smoke, something come from the outside, or just something in the extracellular environment, right? Like inflammatory molecules, Okay, things like that. So think about those as well. All right, you guys know that too. It says revision of the genetic code starts with mutations in DNA. We all understand that we go from DNA to RNA to making proteins. If there is a mutation in the DNA, it can lead to the production of an aberrant protein, right? That then again screws up some cellular mechanism. Okay. All right, so we have to mention tumor suppressor genes again. Now, I know I talked about this before in cell bio, probably mentioned it in bio 111 as well. What do tumor suppressor genes do normally? It says it right there. Slow the cell cycle down, right? It says slow cell replication or cell growth. They are involved in inhibiting the cell cycle, right? Keeping that cell cycle in check so that cells do not divide out of control, okay? It says they may repair DNA mistakes. So some tumor suppressor genes, or genes that we would consider tumor suppressor genes, can be involved in DNA repair, right? Tells when cells should die, also involved in apoptosis, right? We kind of mentioned that on in our cell cycle diagram. Okay, so if there is a mutation that occurs in a tumor suppressor gene and it turns it off, meaning the normal gene product is not going to be made, that then can cause cells to replicate out of control, cause the cell cycle to not be regulated, right, and lead to cancer. This is an example of this p53 gene. It actually also, it interacts with another molecule called p21. Don't worry about this specific example, but mutations, particularly in the p53 and the p21 gene, have been specifically linked to certain types of cancers. And I believe that that's one of the ones that's linked in specific types of colon cancer, right? So there are certain cancers where you can really find a really strong linkage of one particular gene. And so p53 and p21 are examples of tumor suppressor genes that we see connected with a lot of different types of cancers, okay? All right, so there's figure 12.5. Hopefully we understand what this is showing. Yeah? Any questions about that, guys? Okay. Now what about oncogenes? They are the opposite, right? Or what do we call, well, proto-oncogenes first are sort of the normal genes that are there, right? And this is something we went over before as well. So what are proto-oncogenes? What is their normal function? In terms of cell cycles, so let's think about cell cycle. Proto-oncogenes normally do what to the cell cycle? Well, how do they regulate it? Do they slow it down or speed it up? Speed it up. And so when normally would your body want to speed up the cell cycle? Give me an example of a normal situation where the body would want to increase proliferation of cells.
When you get cut.
Okay, good. An injury, right? When you get cut. There's going to be, you know, different cell types that are going to need to proliferate to heal that, right? And basically kind of seal up that tear in the blood vessel. All right. So there are many. There's other examples as well, right? Growth, right? Just in general, cells need to be able to divide rapidly. If you even think about just development from one single cell, right, into the embryo and the fetus, obviously you need to have rapid cell proliferation there, okay? So proto-oncogenes are needed normally, okay? When does an oncogene, or when does a proto-oncogene become an oncogene?
When it's mutated.
Say it again.
When it's mutated.
Yeah, when it's mutated, all right. And then what does that mutation lead to? Does that mutation cause the proto-oncogene not to function? What does that mutation do? Because it's different than tumor suppressor here. We're thinking about this, right? If there was a mutation that turned off a proto-oncogene, well, that would then slow down the cell cycle, right? But that's not what we're talking about here. So if it's going to become an oncogene, the mutation is going to cause what to happen? It's going to cause continuous growth, but why? What's it going to do to that proto-oncogene?
It's gonna keep it like turned on all the time.
Yeah, it's going to keep it turned on all the time, right? Where normally there are mechanisms in place that would turn it down, right? But this mutation that occurs is going to keep it turned on all the time, and it's going to keep making that protein, and it's going to keep stimulating the cell cycle. We understand? So do you guys see how the mutations that occur in a tumor suppressor gene versus a proto-oncogene, or the results of the mutations are different in terms of if they're going to cause cancer or not? Okay, so for the mutation in a tumor suppressor gene, what does that mutation do to the tumor suppressor gene to then lead to cancer? So if we go back, what does it do for tumor suppressor gene? Does it make it, does it turn it on all the time? Does it cause that tumor suppressor gene to be on all the time? What does
No,
it do?
it turns it off, right?
Turns it off, yeah. So if you have a mutation in a tumor suppressor gene that turns it off when it normally should be on, we want to just stay with that terminology, right? That can lead to cancer. For proto-oncogenes, the mutation that occurs is going to turn it on all the time because it normally stimulates the cell cycle. So it's going to keep it on and it's going to keep stimulating the cell cycle, okay? So a mutation in a proto-oncogene that turns it off, would that lead to cancer? No, right? If a mutation occurs in the proto-oncogene that turns it off, that's not going to lead to cancer. It's only those mutations that cause it to stay on. Does that make sense? Okay, so understand that the result of the mutation, is different, or I mean, it can be anything really, right? It just depends. So any mutation that's going to turn a proto-oncogene on all the time will then cause it to become an oncogene. Okay, so an oncogene is not normal, right? A proto-oncogene is. Those are genes that are normally present in our genome that are there to stimulate proliferation, okay?
Dr. Shi,
yeah.
you might have said this, but you said that if there's a mutation that turns it off, is that still called an oncogene or it's called something different?
No, because that wouldn't be causing cancer, right? So you wouldn't call it an oncogene. Yeah.
Okay.
All right. You also, in some types of cancers, in certain types of cancers, we'll see chromosomal translocations occur. So we talked about this before too, and actually, I think we talked about this a little bit even in our genetics chapter earlier on in this course. But this is when a piece of chromosome will break off, right? And then move to another chromosome or something like that. So this isn't just a single, a single sort of nucleotide mutation, right? Or a single gene mutation. This is much larger scale, okay? So these types of abnormalities are very common in leukemias and lymphomas, okay? So one specific one, myelogenous leukemia, and I believe in the notes, I have that sort of defined. When you think about a leukemia, what's a leukemia going to affect? What types of blood cells?
White.
Leukocytes.
The white?
White blood cells. So it's going to affect white blood cells. Lymphoma will particularly affect lymphocytes, right? So also going to affect white blood cells. So there's also, it also mentions this Burkitt lymphoma, so I defined that for you as well. Actually, I think I might have, oh yeah, the myelogenous leukemia is defined on the next slide in the notes, okay? So, so this is kind of just giving you two examples of specific types of leukemia and lymphoma that are caused by translocations, okay? Okay. So if you look, this specifically is showing you for this chronic myelogenous leukemia that you actually can look for this this translocation that occurred in this presence of this Philadelphia chromosome. Okay, this defective chromosome 22. So this is how this particular type of leukemia can be diagnosed. And one of the ways that it's diagnosed, okay, is by the presence of that. All right. So we don't need to memorize this, right? But be aware of kind of what this is showing you. So this is showing you your normal chromosome, right? Normal chromosome 9 and your normal chromosome 22. You basically have a breakage in both of them, and then the piece translocates, okay? All right, other types of, right, so we talked about tumor suppressor genes, proto-oncogenes. We just mentioned that you can have chromosome translocations that can lead to cancer. Also, there are a lot of cancers that are related to mutations in DNA repair genes, okay? And again, I know we talked about this before too in cell bio. And so it says here, although they may be tumor suppressor genes or oncogenes, they're not part of the cell cycle regulatory pathway. So meaning some of these are going to still kind of be, you could still probably call them a proto-oncogene or a DNA repair or a tumor suppressor gene, but they're not necessarily involved directly in regulating the cell cycle. Okay. What about DNA repair? I mean, when we say DNA repair, we already kind of talked about this, right? Meaning if there is some sort of damage that's induced, remember we talked about in cell bio, all these different types of DNA repair mechanisms, right? That like, if you have just a single nucleotide change, if you have a thymine dimer that's induced by UV damage, right? There's a different repair mechanism. So, and in fact, there are particular mutations in those types of repair mechanisms that will repair UV dimers that lead to severe forms of skin cancer, right? And a disease that basically is characterized or hallmarked by these severe forms of skin cancer. So there are specific mutations in DNA repair genes, DNA repair mechanisms that will lead to cancer, okay? All right, so we're leaving this very general. I could have went specifically into DNA repair mechanisms again for cell bio, not for this course, okay? But again, we want to make the connection between this and cancer, right? I could ask you, explain how a mutation in a DNA repair gene could lead to cancer, right? Or would potentially, cause a cell to become cancerous, right? We should be able to make that connection. All right, so kind of just going on here, it says additional genetic alterations. It just says chromosomal abnormalities. So we said chromosomal translocations in particular, but also you do see more chromosomal abnormalities in cancer cells in general. The other example was kind of giving you a specific translocation that occurs in a specific leukemia, right? And kind of talking specifically about leukemias and lymphomas. But in general, you do see a lot of chromosomal deletions in these cancer cells, right? So again, it's just not single gene changes. It's more serious than that. Pieces of chromosomes that are missing, okay? All right, so aneuploidy. What does that term mean? Abnormal chromosome number. So you can have a gain, right, or a loss. So again, remember, these aren't germline, right? Like if you have aneuploidy and it's in all of your cells, we talk about those few, diseases that are like that, right? Like Down syndrome, okay? Here, we're talking about on individual cell level. So if you're looking inside that tumor at specific cells, you'll see that many of them will have changes in chromosome number. They'll have abnormalities in the chromosome, okay? So remember how that's different. All right. Also, these cancer cells can amplify genes. So they have the ability to kind of just increase the number of gene copies. So say like they want to proliferate and divide a lot, right? They can make more of a protein that's going to stimulate the cell cycle just by amplifying those genes. Okay, so that's another sort of abnormality that you see. So there's lots that we listed. More things that are involved, growth factors, okay? This think of as just, so this is another kind of, it's a little not quite right, but the idea is just understanding that if there's a defect in a receptor that responds to a growth factor, that's an abnormality that you see in cancer cells as well, right? So it's the kind of, there are particular types of receptors and proteins, these RAS proteins that bind to specific receptors, and those have been implicated in cancer. And we consider them proto-oncogenes, okay? Because they normally will respond to a growth factor. So what does a growth factor do normally?
It causes the cells to divide.
Yeah, it causes the cells to divide. So it's something present in the extracellular environment, right? That's often, it's secreted by other cells, right? And if it's there and it binds to a cell, you know, like on one of these receptors here, it will stimulate that target cell to divide, right? So do you guys see why you might consider a growth factor receptor as being a proto-oncogene, right? Because it's there to basically stimulate growth and proliferation, okay? So again, I think here what we really are getting at here is the idea that growth factors and receptors for growth factors are going to be connected to cancer, right? And if there are changes in the amount of growth factor around or in the receptors, it can it will alter the growth of that cell and thus lead to cancer. Okay. This last concept here, I had to kind of like read over it again to kind of like get it in my mind. So again, remember, you're thinking about like in a tumor, like within a tumor, right? Looking at those different cells there. You know, you may see some cells that have like one normal allele and some cells that have one mutated allele, right? So you have one normal copy, one mutated copy. Those cells are fine because they still have that normal copy to compensate, right? Remember, we always have 2 copies of every gene, right? We have 2 chromosomes. We got a maternal and a paternal allele, okay? The idea here is that, you know, if a second mutation occurs that then affects that normal allele, that cell will then become cancerous, okay? So that's why it's saying you lose that heterozygosity, right? When we say something has one normal and one mutated, we consider that heterozygous, right? And so that's where this term is coming from. So that's the thing too, like if you look at a tumor specimen, you can actually see this mix of tumor and non-tumor tissue kind of because of this. So there were some that just had this one mutation, but then as more occurred, you see some cells that have both and then therefore are cancerous. Okay, so that's kind of where that idea is coming from, or that term there. Okay, so let's talk about this multi-step process of carcinogenesis, okay? Talk about angiogenesis a little bit more and some other specific mechanisms, I guess, okay? So these are 2 models, I guess, of cancer evolution that are out there. I don't know that they like, I guess kind of things can happen in combination of these 2, okay? But these are kind of 2 just different ideas, and I'll show you a picture, it'll make more sense. If you talk about the stochastic model, it's the idea that each cancer cell has the ability to proliferate and form a variety of new tumors, right, from the same tissue. So meaning like every cell that's there kind of has this sort of cancer-like ability. As opposed to this cancer stem cell model where it's saying that most cells have a limited ability to proliferate, right? But there's only a few of these stem cells that exist kind of in a tumor. And those are the ones that are going to proliferate and divide. So it's not every single cell there within the tumor, okay? The other thing here, and I mentioned this before, it says here, cancer stem cells are insensitive to chemotherapy and radiation because they don't rapidly divide. It's a little misleading, that statement, so I felt like I had to kind of clarify that. So remember I just mentioned the kind of that G0 in the beginning of class? And like when you think about remission, so if these stem cells that are going to be the ones that will proliferate and divide go into G0 in like this resting state, and kind of then the patient would probably be in remission at that point, giving chemotherapy at that point is going to do nothing to get rid of those stem cells. So that just means they're hanging around there, right? And then when there's that environmental trigger or that inflammatory trigger, whatever, they then can enter back into the cell cycle, and then you start to see formation of tumors and more cancer cells produced, okay? So I guess the idea is if all of the cells kind of were always in the cell cycle, and you didn't have a few that weren't, it would be easier to kind of get rid of them, I think, right? But because you sort of have differences, it makes it a little bit harder to treat. So this is just showing you here, like here's your cancer cell that has the ability to divide. All of them have the ability to divide and basically produce more cancer cells. So like kind of all of these are the same. Here though, you're only going to have certain cells that can actually produce a cancer cell. I think they're saying green. Yeah, so this is showing you here, like in this cancer stem cell model, you only have a few green cells that are cancer cells, okay? Whereas the other ones appear normal or act sort of normal, Okay, so it makes it a little bit harder to treat when you have this sort of diversity of cell types within, if you think about just within a tumor. Okay. All right. So we mentioned this angiogenesis. Why is it important? Obviously, if cells are rapidly dividing, they're going to need a supply of nutrients, right, and oxygen, and therefore important for them to get new blood vessels to that area. Okay. So it says here, as tumor cells grow and divide, the resulting cellular mutations restricts oxygen supply, and that can lead to areas of hypoxia. And so, right, meaning you have a whole bunch of cells that are dividing, there's eventually going to be a sort of shortage of oxygen. Normally then, those cells should just die off, but they don't, right? Because they have this sort of, they have the ability to go through anaerobic glycolysis, right? They'll produce lactate. And obviously you're not going to be able to produce as much energy as you can, but just by producing that lactate and that decrease in ATP production, that can actually lead to fatigue, right? So often when you think about cancer symptoms, fatigue is one of them. So as those cancer cells are forming, and it kind of think of like your body's energy is kind of going to these cancer cells and giving them the ability to divide. Okay. Now eventually this kind of will run out, right? Like I said, then they have to then produce more blood vessels to get more nutrients there. Okay. So that's what this is now talking about here. And actually there is a particular growth factor called VEGF that causes growth of new vessels. Okay, so like that growth factor is necessary for these new blood vessels to form. So it's an important molecule when you talk about angiogenesis. Okay, so you look here, it's showing you there, right? That tumor, those tumor cells, those cancer cells actually produce and secrete that VEGF themselves, right? So they're helping themselves out. They produce this growth factor that's actually going to lead to angiogenesis, the formation of new blood vessels. Okay, and now once this smaller tumor up there now has access to more of these blood vessels, it can grow larger. And then what else is probably going to happen now? There's going to be more blood vessels that are in closer contact with those cancer cells. Once one can break away, it can then enter into the blood vessel, right? And then can go wherever. And then that's when you talk about it being malignant, right? It can basically move to other areas of the body, okay? So I think you guys could, if I ask you a question like, what would happen or what would be the result of the sort of, if the cancer cells weren't able to secrete VEGF, right? What would be the result of that? So say you have a tumor, those cells in that tumor are not able to produce VEGF. What's that going to mean for that tumor or those cancer cells? Yeah, they probably won't grow as much. Why? Yeah, well, what can't happen there in that case if there's no VEGF? Yeah, new blood vessels aren't going to be able to be formed, right? And then, right, yes, that will limit their nutrient availability, oxygen availability from there, okay? So I guess there's kind of two things on here. This first bullet point talks about, the idea that these cancer cells need more oxygen, right? They need increased amounts of ATP. And so the fatigue that often accompanies cancer is a result of this, right? Then what do those cancer cells do to sort of override that lack of oxygen? Well, hey, they start to produce VEGF and they get more blood vessels to their tumor, right? So they can keep growing. Okay. All right, so I think this, I don't need to read what's on here, but this is really talking about different growth rates, right? Some tissues, growth rates are rapid, okay? Other tissues, slower. Really just kind of based on the sort of innate characteristics of that tissue and those cells that exist there, okay? In terms of time spent in the cell cycle, it says there's lots of things that can influence that time spent, okay? The pH of the cell, how much oxygen is available, how much glucose is available, okay? All right, I think we kind of talked about most of this already. Right, so remember that if there is a growth factor that's there, right, it binds to the receptor, it then activates a signaling pathway inside the cell. That then does what? Ultimately, it will basically cause the cell to produce more proteins involved in growth, right? And survival, and cause that cell to be able to keep surviving, and then obviously proliferating as well, okay? All right, so in terms of this multi-step process of carcinogenesis, let's look at the figure on the next slide. But there are 3 steps, right? 3 main steps here: initiation, promotion, and then progression, all right? So this figure basically describes those 3 steps, okay? There's some cancer-causing agent that might damage the DNA of a cell, right? The cell's unable to repair that. Okay? You end up with some permanent mutation to the cell, okay? Now there comes some other factor, right? Some other carcinogen that sort of promotes or increases that DNA damage. And then you basically have this mutation that cell that then is able to kind of start acting weird, right? And doing things that it's not supposed to do, can start then producing more cells like it that then can lead to the progression and the formation of a tumor, okay? So the words on the slide before this basically just give you some description of what's happening in the pictures there. Questions about that, guys? All right, so this is the last section. And like I said, the recording for part 2 picks up with section 12.6. And I think it's the epidemiology of cancer. That's where it's going to pick up with. So let's finish this quickly, so then that way there's no gap in what I covered. All right. I think this is, okay, so this is something we haven't talked about yet, right? It says altered cytoskeletal control, okay? So what about cytoskeleton? What does the cytoskeleton do? In normal, right? Normally, what's the cytoskeleton there for? Structure, right? Movement of things through the cell. If you think about microtubules, which are part of the cytoskeleton, they're particularly responsible for separating chromosomes during cell division, right? If we did mention that in cancer cells, oftentimes there are, a lack of chromosomes, right? Or an extra chromosome. Why? Because perhaps something's going wrong with the microtubules. They're not able to separate those chromosomes like they should. But it also, so that's one thing that's involved in, but also cytoskeleton, right? There's proteins that link the cytoskeleton inside the cell with the extracellular matrix, right? And basically anchor cells, okay? What happens with cancer cells is that they lose that sort of connection to the extracellular matrix and the anchorage. And so they lose the contact with other cells, like I said, and that extracellular environment. So normally what will happen is something called contact inhibition, right? If a cell is basically in contact with another cell, it tells it, well, maybe I shouldn't keep dividing, right? It sort of inhibits its growth. But cancer cells lose that ability. Okay, so their cytoskeleton, the extracellular matrix, all these proteins that are sort of involved in structure and anchoring cells are again, they're not working properly. Okay. Yeah, so again, glycoproteins, glycolipids, all of these proteins that sit in the cell membrane, again, are not functioning properly, or they're not linking with the cytoskeleton and the extracellular matrix, okay? One particular protein that's in the extracellular matrix is fibronectin. So you do see that there's a loss of fibronectin in these cancer cells, these malignant cells, okay? Again, that's a protein in the extracellular matrix, okay? And so this is what this is showing you here in terms of kind of just following this along. You basically can see now how these cells, again, they lose contact. So if you look at that basement membrane, they would normally be anchored there, right? Again, they would be connected to the extracellular matrix in some way that keeps them where they're supposed to be. But they lose that. And then they're allowed to, or able to break away when they normally shouldn't, right? And then what happens? Now it's getting into a blood vessel. Now it can go to different sites of the body, right? And form new tumors, okay? So if they didn't lose that ability or that grounding or that anchorage, they wouldn't have this ability to move again into blood vessels and move to distant spots, okay? This is the last Yeah, last thing. We're just about done. So in terms of, if you kind of think about patterns and how tumors spread, right? You have primary tumors. Those are the first tumors that form, okay? And they actually will have the tendency to metastasize to specific organ sites, depending on where they start. So that gives clinicians, you know, if you talk about breast cancer, a lot of times breast cancer will then, those tumors can metastasize to the lung. That's usually the first place. Also to the brain as well. And so if somebody has breast cancer, you're definitely going to get a scan of the lung, right? And probably a scan of the brain to make sure that metastasis hasn't occurred. Okay, so there are particular kind of sites where things will go. And so that's what this table is showing you, right? You see the primary tumor site, and then the most common metastatic sites, right? Okay, so important that clinicians know this, right? Again, you want to be able to make sure that it hasn't metastasized. Okay. I have a friend who recently passed away from breast cancer, what, a year ago? Maybe it was a year and a half ago. And she went, I have probably told you guys this story before, right? Like she went back and had a scan actually of the lung. And at that point, they didn't see anything there, right? So she thought she had a double mastectomy and everything and thought she was going to be okay. Like, right, that was good news. But then it turns out, you know, later on, it did end up metastasizing in the lung. They just weren't able to see it at that point. And then she ended up with brain cancer as well, right down the line. So they always will look in the lung if following, you know, breast cancer. All right, so some of these types of proteolytic enzymes here. So these Adams proteases are a specific type of proteolytic enzymes. Basically, they degrade proteins, right? So they're secreted by these cancer cells, and they can basically degrade those structural components of the extracellular matrix. So they're kind of like chewing away at whatever was holding them down, and that then enables them to be released and enter into blood vessels and metastasize. Okay, so that is pretty much it. The recording, the part 2 picks up right here. So I went through the last couple of slides a little bit quickly. So make sure, again, you guys look this over. I will post this recording, obviously, so you can still go back and listen to it if you need to. All right, guys. So if you don't have any questions, bye. I'll see you next week. We are virtual both days next week. Okay, guys? I'll stay on if anyone has questions. Let me stop recording though.
Bye, Dr. Kaminsky.
Bye, take care.
You too. Doctor.
Oh, wait, 1 second.
Okay.
Why did that not? Oh, did it stop share? Yeah, duh.
It did.
Never mind. Let me stop the recording. There it is.